So this week, we'll be covering just some of the basics of 3D and 4D visualization using Imaris and Vision 4G. Really, this is more of an example of the kind of software that the core has access to and encourages you to use. So we'll go over a few basic examples of how visualization works in both of these and what strengths, maybe weaknesses, each of the pieces of software has. So we'll go forward from there. It's the same set of introductory slides. I'm Stoyan Ivanov. I specialize in data analysis for the Integrated Cellular Imaging core here. At any point, you can come see us physically or send us an email to request a consultation. Consultations are always free. So you can come to us with any kinds of questions, concerns, any issues you have with the software or our microscopes as you're using them. All right, we'll skip through this because I imagine most of you have seen them. But what is the purpose of today's talk? Well, as I mentioned, I want to introduce you all to Imaris and Vision 4D and show you just the basics of how they work. We'll go through a couple of examples in each of the pieces of software. We'll see how they function, what the user interface is like, and how what situations you might prefer one over the other. So what are these? Well, they are visualization and analysis software suites for large data sets. And what does that mean? Well, that means you can get a very nice, very detailed Z-stack of something, and then you can import it in one of these pieces of software and start analyzing it. And what does that mean? That could mean denoising in part. That could mean identification of certain physical structures in the data that you're interested in. You might be trying to calculate, the total volume taken up by a certain kind of cell, or track a dendrite from start to finish and then calculate quantifiable statistics. How big is it? How many times is it branched, etc. So that's the kind of really computationally intensive analysis that these softwares can do for very large data sets. Both of these are licensed pieces of software. So the core has a license that allows a number of users to use the software. Specifically, we have several licenses for Imaris. So at any point, one of you can log on to one of the workstations listed for Imaris and work on it concurrently with someone else. But Vision 4D currently, we've only got one license for. So at most, one user can use Imaris 4D at once. So that's a little perhaps harder to access. But yeah, at any point, you can contact me if you have any questions about either of these softwares, whether it's applicable to your kind of analysis, whether you just need some help with it, or you have more broader questions, let's say, about what it can be useful for. And I don't want to forget to mention that there is also a visualizer, which is free from Imaris for visualizing either data you've already performed analysis with Imaris on, or just maybe raw data from microscopes. So that's something you can open up and install locally on your own PC, your own laptop, your own Mac, and at least take a look at data there, start the process of visualization, even if you aren't able to do any of the more nuanced analysis. So how are they the same? Well, we covered a number of these things already. They can be applied to similar types of research. They can be applied to similar types of shapes or structures. They can be used to track neurons or walk along the path of a filament. They can be used to track the movement of cells or bacteria with a with a tracking function embedded in there. You can do all sorts of things like create surfaces of the various structures, distance maps, and get data on how certain structures are, how far are they from other structures of interest. Perhaps it's, how far are these cells from this filament I traced, and that filament is actually tracing a blood vessel. Questions like that. Below, you can see an example of one of these fits of a filament tracing of a neuron. And we'll actually do an example that involves filament tracing. And we'll get to see how you can go from data to an example like in the picture shown below. Yeah. The secondary thing which I want to stress as well is, in addition to creating these structures and performing these analyses, which are very nice for visualization, what both of these softwares also do is they are able to provide quantities. They're able to provide numerical values that can get you a volume of the relative structures, that can get you separation distances, all of those things that would normally be very tedious or impossible to do for a three-dimensional data set by hand. Using this software, you can then quantify it a lot more comprehensively in order to perform your analysis and analyze your data effectively. Now, all of these calculations I'm talking about, all of the statistics that you would get from both of these softwares, does beg the question of how taxing are they to run for a computer. So we'll take just a very quick aside. I won't go over all of these terms in detail, but we want to remember latency, which is how long it takes a piece of data to know that it's being moved somewhere else or being analyzed. And just RAM and hard disk space. Those are two of the big ones, in addition to GPUs and CPUs of the computer. So yeah, commonly these softwares load the data into RAM, so it can be easily accessed. And what does that mean? Well, accessing something from a hard disk, for example, is akin to sending someone a letter. Accessing something from RAM is much, much faster. It's akin to sending someone an email. So that question is important because it means that if you were running one of these pieces of software on a computer that didn't have the components to run it effectively, you'd get slowdowns, and it would take a while to analyze, to create the surfaces, to create all of those things that you're interested in doing and analyzing. So with that being said, the core, the ICI core, has workstations that have been built specifically for these kinds of softwares. So if any of you install the viewer for Imaris on your local computers, or you get a trial license for these softwares, its performance on a local commercial computer may be very much inferior to how it would behave on one of our workstations, because our workstations are designed for that kind of software to run effectively. Yeah. Now we'll move on to some differences. Imaris generally, in my mind at least, has a lower barrier to entry. the analytics you can do with it, the visualization, is generally much easier to access, with the caveat that it's less flexible than Vision 4D. Vision 4D, on the other hand, is very flexible. You can do a lot with it, but because you're given that freedom, it's also much more difficult to jump into. A secondary but very important consideration is how both of these programs are able to parallelize processes and how you can build a pipeline in each of them. Now, parallel processing, I've included two images here to illustrate that. When you are able to parallelize a method of calculation, you're essentially able to break it up into tiny little pieces that are all calculated in the same way. They don't affect each other. And so you can give that out to the various central processing units a computer has. So if you can parallelize something, send it off to a CPU. If that CPU has a lot of cores, like our workstations do, then you can analyze it much more quickly because all of the parts are being calculated at once. Vision 4D does a better job of performing calculations in that way than Imaris. Imaris is generally less parallelized. So for calculations like the neuron or filament tracing calculation, those can be done much more quickly with Vision 4D for a very large data set than they can be for Imaris for a data set of similar size. And that just comes to how those processes were implemented in each of these softwares. And the last thing I mentioned is pipelines. Say you want to perform a certain function that includes, or a certain analysis that includes a little bit of machine learning or a very specific kind of analysis, in the same way that we're able to batch in Fiji with macros to perform the same analysis on a bunch of different data sets, you can do the same thing with Vision 4D, where you have a pipeline of analytical steps. You perform a data set, and then you can perform that exact pipeline on a bunch of those sets. That is harder to do with Imaris. Usually you'll have to go through one by one, but Vision 4D is made to be used in that kind of batch analysis way. So if you have a lot of calculations that you need to do to a bunch of different data sets, Vision 4D is generally pretty good for that. Yeah, and here we will go through a set of 2 samples. Since the meeting today is only for about an hour, that might very well take the rest of the session to go through, but we will look at surface creation or structure identification in both of these softwares, and then we will also do a quick filament tracing step with both of these softwares to see how that works and what that looks like and how it compares, just as an example of the kind of stuff both of these softwares can do. But before we go into that, does anyone have any questions so far? All right, let's go. So Gaurav, can you see my screen? Well,
yes, everything
okay.
is good.
Wonderful. So let us go back here. So this is the Arena tab for Imaris. when you observe a folder, you can see what data sets are inside. Both Imaris and Vision 4D use their own proprietary formats, and they store the data that you have, as well as any statistics or calculations or object creations that are done in situ to analyze that data. But they will be in that .ims file type, and they don't usually play nice with something like Fiji. So that's just so you're aware. But we can import one of the data sets here and view it three-dimensionally. If we click the left mouse button on a PC, by the way, all of our workstations are PCs, just to consider for anyone who might be more used to Mac. We can visualize the data three-dimensionally. We can change and enhance brightness that we're interested in. We can even, let's do that, crop a subset of everything, of the entire data set. So say we only wanted to fit this central blob and nothing else. We want to select that section and then crop it, and then we'll only see that section. This is less important for a data set of this size, but it can become much more important when the data set is very, very large, because when you're performing an analysis like this, it will be applied to your entire region of interest. So if you're looking at the entire data set, it could significantly prolong the amount of time it takes to finish the calculations to run the various algorithms. But in this case, let's say we wanted to know the volume of the structure. We've got the volume here, and that's basically just our data. Using the display adjustments here, we can take away or put back our various sets of data, but we're interested in creating a structure, or a surface rather, a surface that will cover the outside of the structure. So we can go here to add a new surface. If we double click on the name, we can rename it. Generally, it's good practice to name your surfaces or structures or your analytical steps in some way so you can keep track of them. I know I've forgotten more than once what I was doing to which of those surfaces, so it's generally good practice. Initially, it will take you to this automatic creation wizard, which is good enough for us. We can uncheck classify surfaces and object-to-object statistics because they're not going to be useful for us right now. In principle, those two options can be very useful if you're comparing several objects to each other or to something else in the data set, like how far away are these cells from this other structure that I've identified. That's how you can calculate questions like that. That's how you can answer questions like that. You're prompted to pick your source channel. So we'll pick channel 1 at 435. Smooth indicates that the surface will be smoothed. It'll be continuous. And the surface details indicate what the smallest amount of surface the program will look at will be. So that the lower that value is, the smoother and more continuous the surfaces you generate will be at the cost of processing power. Very smooth surfaces will require, with very smooth surfaces with very high resolutions will require a lot more computational time to render and analyze and calculate the statistics for than sort of rougher surfaces. So finding a good balance between fidelity and roughness depends on what you're analyzing and what you're trying to get out of it. But anyway, we're taken to this option where we can modify how expansive our surface is at the far ends. We generally want to stay somewhere in the middle. Something that encompasses the signal we're interested in, but doesn't over exaggerate how large the structure is. And something that doesn't under calculate how expansive the structure is. So you see here, we're leaving a lot of data unincluded, or a lot of data is not included in the surface that we're generating. But we also don't want to go too far and include much more volume than we think we need. So near the center here, we have a surface of that structure. We can continue forward. Calculate the surfaces. And right now we're taken to a second adjustment panel. And here we can actually filter out surfaces which are too small for us. For example, this one here, or here, we can filter those out and retain just the central one we're interested in. Click to finish. And we're left with a surface over our data. Then if we go to this panel right here, we can get all sorts of statistics. How large is it? What's its volume? We can export these if we want. We can analyze them later as we need, but all of that is available here. And we can, in fact, configure what kind of statistics are displayed through the menus in the software. So you can get pretty granular in what kind of numbers the software outputs. Now, let me show you the same thing in Vision 4D. Just for a quick reference, Vision 4D, Imaris 9.72. That's one of the latest version of the software. Generally, if you can, you want to use the most modern, the newest version of both of these. And this is just the desktop on Workstation 0, so you can access Vision 4D here, Imaris 9.72. And in fact, in the near future, they are releasing a newer version of Imaris 9.8, I believe, that includes improvements to its performance. So that's something to check out if you find it interesting and potentially useful. So we can open one of these files and take a look at it. This is the same file we just looked at. Excuse me, the same file we just looked at. The UI is different for Vision 4D, which may turn some people off, but you can look through the layers of the stack, the slider up here, or you can show as a 4D viewer. Take a look at it in 3 dimensions. For what we're doing right now, though, it's easier to look at it in the 2D fashion. And the first thing you'll notice is this analysis pipeline tab here. You get to it by clicking here, Add Analysis Panel. And that is the set of steps that Vision 4D will perform to the data. We can define an ROI initially because this is a relatively small data set. I'm not going to define a smaller ROI. In Imaris, we did define just the central region. Here, I'm not going to do that, but you can, if you need to, crop it in the same way. You can zoom to this, then enter custom. Determine which channel you're cropping, so it's this one. Then if we click down here for the bounds, we can specify just the bounds we're viewing. So right now, the square that we can see, that's our defined ROI. But we want all of it. So that would in fact be, yeah, current image set. That would be the entire image set. When we're ready to move forward in our pipeline, we can go click next operation. That will do some background correction. We see that process running here. Okay. Click forward again. Now we're segmenting it. And that might take a little bit. So let me show you what I was talking about earlier. These are all the logical processors in this workstation, workstation 0. And when you see each of them showing a lot of use, that usually indicates that a program is running a calculation or an analysis in parallel, like they're all working, they're all doing an analysis. So if you happen to be using a computer and happen to be curious, that's one way to view whether your software or your algorithm is using more than one core at a time. Mhm. Okay, let's let it finish.
So, and while it's trying to finish, I want to bring up something right where you were in Imaris, which is it's good for users to import their data from cloud drive and store it on the data drive of the computer and then use Imaris versus trying to work directly from the cloud drive, because that's gonna all depend on the connection, and it slows down the processing. So if you want to highlight that part while you're in Imaris.
Yes, that's exactly right, Gaurav. So any data you capture from one of our microscopes gets uploaded to our cloud drive. Usually it's under the letter Z for ICI Cloud. Uploading and downloading to that cloud takes time. It takes time. So a large file that's over 500 megabytes, if it's a few gigs, it can take a while to download and upload. If you're trying to run the software on a file that's on the cloud drive, not only is that going to slow you down immensely, because now you're adding a lot more latency to the process. If writing a letter and mailing it was what you're doing, if you're reading something off the hard drive instead of from RAM, then this would be like sending something in a chip across the world. It takes a lot longer to do, and it's a lot more prone to errors, permissions errors, among other kinds of errors. So download what you want to look at locally on the workstation, look at it there. It'll be much faster, more reliable. And then once you've done your analysis, upload it to your OneDrive or put it back on the cloud somewhere where you can fetch it. All right, that was a good aside, because now we've got the finished analysis here. If we go through the different slides in the stack, we see that this has been applied to all of the data. And from here, we can take a look at these statistics that we got. There we here. In the same way that Imaris, you could modify what kind of statistics you get shown. You can do that here as well. So let's add volume. And then we can see a breakdown of all of the segments we've created through the various layers. And then from those, we would be able to calculate, say, a volume for these structures. We'd look at them and be able to say, how well do they fit? We can already note an issue or two here. The way we've done it right now, we are missing some of the fitting here and here. It looks like whatever we specified our parameters to be, they weren't quite good enough. So we would go back and we'd redo the analysis until we got all of the things that we were interested in and really said, this is done well now. So that's a kind of iterative process that, using these kinds of tools, you might go through several iterations to find a good model or do the right kind of deep learning on your data set to make sure that the algorithms in the program get you the results that you are looking for, or analyze it rigorously and confidently. So that's example one. Just something quick before we close out of this example. These pipelines, you can save them at the end. You can import them and export them And then later on, you can open up another Vision 4D instance and then run it there as you need to for new data. So you're not going to have to rebuild this every time you need to use it. You can import it, manipulate it as it's needed for your new data set, and just build off what you had done last time. All right. Now we'll take a look at the filament tracing function. So in the same way we did before, we'll open a data set, and this is quite a big one. Quite a big one. X, Y, and Z. So we can see a lot of cell bodies here. A quick note, the rendering quality slider on your volume component determines how well Imaris displays the data that it's looking at. The better the quality, the more expensive it will be for your system to analyze. And if your hardware isn't specialized enough, you could start to see slowdown. A partial solution is to reduce the rendering quality, and that will reduce the rendering quality. As you can see now, it's important to note that while it looks messier and less good as data, this is only changing what you see on your screen. It's not actually altering the data that the software is looking at. So this would be a superficial change. The analysis could still carry on underneath. So this is very large, so we want to actually crop this. So let's go to Edit, Crop 3D. And let's just focus on one in the center here. We can see in XY. Okay, we've got just this component. So we'll go to New Filament. We will follow the automatic suggestions. The values here, largest diameter and thinnest point, these are actually calculated by the software based on the data it's looking at. So they're variable. And if you find that these two values aren't ideal, though they're usually pretty good as starting points, you can adjust them manually right now. But we're happy with the automatic ones. Right now it's populating the volume with a bunch of seed points, including the cell body here. We can take a look and see what it's like. And you want them to be labeled well, but you don't want too many, because if you have too many of these seeds, not only is it going to take a lot longer to process, it might not improve the quality all that much in the end for the additional time. So if you see some that are now obviously not something you want to keep, like for example, we've got these two seeds that are right next to each other. So we can probably do without one. You would press Shift and left click and remove that from there. So let's go automatically and reduce the quantity. We're happy with the seed point. It's right over the cell. We can even include further specifications like removing disconnected segments, in which case only the segments which end up going back to the big seed point will be retained, everything else will be thrown out. But for the purposes of this exercise, let's not check that box. And then we run the calculation. As you can see on this processor here, this is a serial calculation. So that one processor is going to do all the analysis, which means it's going to take a little bit. This is one of the slower functions in Imaris that we've had users say, oh, I don't know what's happening. The calculation is going by so slowly. That's because of the nature of the method that's being used to calculate the filament lengths. And that is why we ended up cropping the much larger data set, because that data set would have taken 15, 20 minutes maybe to run one of these on. And until we say have a better idea of what the data looks like, it might be better to stick to a subsection where we can play around before we're ready to commit that kind of time investment. So the process at this point is similar to the previous one I showed you, which is we want to capture all of the signal we can without capturing too much. So we don't want too much green to show up as we're looking at this, but we also don't want no green to show up. We want to be right on the edge. So that looks good. So we can continue and finish. That's interesting. That's odd. For some reason, it wasn't showing up previously, but it would. This is one where I've run that algorithm on the whole volume. Yeah, that's kind of what it would look like. But yeah, it's bizarre. So we can do that in Vision 4D as well. If you take in your data like before, you can do the entire analysis using the analysis pipeline. So you can actually go back and undo calculations you've done. This one we're going to do for the entire data set. So I can show you directly the comparison between Imaris and Vision 4D. Because Imaris is going to do it, or Vision 4D rather is going to do it in parallel time. So you can see, whereas Imaris was using one processor to do the calculations, Vision 4D is using all of them. So this is going to go a little bit faster. In fact, there's 32 cores. It might go 32 times, up to 32 times faster than Imaris for the same kind of calculation, because it's being spread over the entire system instead of just one of the 32 cores. Yes, but while we're waiting for that to calculate, does anyone have questions, or does anyone want me to focus on something specific right now?
So is it possible to do some branch point analysis for the previous data set in Imaris? When I say previous, the same one. But if somebody wants to look at dendrites branching out from one another and figure out how many branches are happening, can we get those kind of statistics?
Yeah, that kind of calculation. Yeah, you can do that kind of calculation with Imaris pretty well. So let's go through that again. Mhm. There it is with Vision 4D. Same kind of calculation. And yeah, you can save that data where you need to. You can take a look at the statistics as well.
So I just want to bring this point down. When we look over Zoom, the rendering does not look that good, and that's because we are on a remote connection. Otherwise, when you're on the workstation doing this personally, it's pretty much flawless.
Yeah, definitely. Thanks for bringing that up. Because yeah, any kind of remote work, which is what I'm doing right now, I'm remotely accessing Workstation 0 from the Whitehead building. That is going to degrade the apparent resolution or apparent quality of the image. But all of your statistics, all of your analysis is still going to be done on that machine where you're running the software. So it's a lot more flexible that way. Unfortunately, you might have to deal with some stuttering or a reduce in apparent quality when you're working remotely. You might have seen once or twice the entire volume we were looking at was spinning, because that is, yeah, a result of the latency in communication. Okay. Mhm. I'm not certain why it's not actually showing up right now. But yeah, you would be able to do the analysis by pulling the statistics from here. If we could figure out why it's not showing. Yeah, I'm afraid I don't know what's going on with Imaris right at this moment. But if any of you are curious, reach out to me and we can do a consultation. I can take you through it a little more in greater detail and take a look at any of the data that you need looked at. Okay. So yeah, sorry, Gaurav. I don't know that I'll be able to demonstrate that right in this moment.
That's okay. We can do this later on. If somebody has a question on that, we can do it in person with them.
Definitely. And again, don't hesitate to write Gaurav, write me with questions, or you can feel free to ask them on our Slack channel as well. Okay. Does anyone else have questions? Would anyone like to see something specific?
I have a question.
Yeah.
So I've been using Imaris and I feel like it's not representing my data well. Like I use the filament tracer. I feel like it's not tracing it as well as it could. So do you have any advice on how to get it to be more accurate? And then also if you section off a small area, how do you then apply what you sectioned off to the whole image?
These are very good questions. We'd have to take a look at your data to see exactly where the issue arises. Without doing that, if when we specify the number of seed points, if you specify too many before running the algorithm, that could take a while. It could also give you, not ideal results. Changing that value will may affect how accurately the filaments algorithm fits your data. So those kinds of questions, whether even the filaments algorithm is the best one for your data, depends on what it looks like. So it's something we'd have to do a consult for, exactly what those are for.
Okay, thank you.
Yeah.
And Gabby, out of curiosity, I'm sure your data set is from neurons, but in that situation, when you are imaging, is that at the best Nyquist sampling? Because that is important, right? For example, the data set that Stoyan is working on right now is at the best Nyquist sampling. And that's why every single fluorescent source is connected, or fluorescent pointer voxel is connected to each other. So typically, sometimes you want to acquire some data set very fast. What you end up doing is you end up saying, okay, I want to acquire data set every 1 micrometer apart versus every 0.2 microns apart. And that is where some of this sampling issues happen. And that also helps with the connectivity issue that you might be having.
That makes sense. Thank you.
And I will also go ahead and link a paper directly to the chat. And that would just be an example of an instance where the filaments tracer could be a good option to start analyzing that data, because you've got filaments in that paper, they're vascular, but they're still organized in that kind of way. So that would be definitely something you could apply that tracer to track how long the blood cells are, what the internal volumes are, how many times they branch, and things like that.
Sorry, go ahead.
I just want to mention that paper is courtesy of Shweta. It's not us, so that's all you, Shweta.
And I was just going to ask him that, yeah, I have read that paper, and in that paper they have actually used ImageJ, but do you think like Imaris is better option than ImageJ because they have used that for the analysis?
It really depends on user proficiency and skill. Imaris is very good for visualization, and it can be very good for analysis. Which is not to say that ImageJ or Fiji is bad for analysis. It just, how much experience do you have with the work? How easily can you look at it? There's some times where you might want to do analysis with Fiji if you already have a way to automate it or way to analyze the data, but lacking that, you might just use Imaris to visualize it, to play around with it before you get to something like writing a macro or a more intensive analytical method with Fiji that might require more time and more technical knowledge than Imaris might to begin with. So they're both good in their own way, is the answer.
It basically depends upon the user, like how well or the user can tackle with either of the software, I guess.
Mm-hmm. Yeah, exactly. Any others? All right. Well, then I think that concludes today's meeting. Again, contact us by